Good morning, we are the team number 4 and today we are going to talk about one of the topics of stage 2, obtaining energy, photosynthesis and respiration. The team is conformed by Alejandra Guiñaga, Ashanti Garcia, Juliana Hernández, Carmen Hurtado, Daniela Ramos and Valeria Vargas.
Photosynthesis. During photosynthesis, plants take in carbon dioxide and water from the air and soil. Within the plant cell, the water is oxidized, meaning it loses control, while the carbon dioxide is reduced, meaning it gains electrons. This transforms the water into oxygen and carbon dioxide into glucose. The plant then relays the oxygen back into the air and stores energy within the glucose molecules. Chlorophyll. Inside the plant cell are small organelles called chloroplasts, which store the energy of sunlight. During photosynthesis, chlorophyll absorbs energy from blue and red light waves, and reflects green light waves, making the plant appear green. The process of photosynthesis is broken down into major stages. The light-dependent stage, also known as the Calvin cycle, takes place in the stroma, the space between the thylakoid membranes and the chloroplast membranes, and does not require light, hence the name light-independent reaction. Energy from the ATP and NADPH molecules use the same carbohydrate molecule like glucose from carbon dioxide. The light-dependent reactions take place within the thylakoid membrane and requires a steady stream of sunlight, hence the name light-dependent reaction. The chlorophyll absorbs energy from the light waves, which convert into chemical energy in the form of molecules ATP and NADPH. So, the cellular respiration in general is the process that occurs in the cells by which the chemical bonds of glucose are broken to obtain metabolic energy in the form of the ATP. The glucose is very important. It's used to a raw material which is metabolized for the dioxide of carbon and water, and this produces energy. The ATP molecules are the source of energy that is used to default to carry out the cellular metabolism. They are formed to the adenine, ribosome, and 3-phosphate groups. The cellular respiration have 3 phases. The first is the glycolysis and takes place in the cytoplasms, and the Krebs cycle and the electron transporting change. These both phases take place in the mitochondria in the presence of the oxygen. Okay, the first step is the glycolysis. In this process, is the conversion of glucose into the 2 molecules of fibric acid, and 2 ATP molecules are used, but are produced. The hydrogen, together with the electrons, bring to coenzyme called nicotine and adenine nucleotides and forms NADH and occurs in the cytoplasms. The citric acid cycle, also called Krebs cycle, is which the breakdown glucose is complete. The glucose molecule is completely degrading on the 2 piruvic acid molecules enter the citric acid reactions. This cycle can degrade substances other than acetyl-CoA A, such as breakdown products and lipids and proteins, which enter the different points on the cycle, and energy is obtained. The electron transport change is the final step, and the citric acid of the dioxide of carbon has been produced, which they eliminate and the molecule ATP. However, most the energy in glucose is carried by the NADH and the FADH2, along with their associated in the electrons. One electron transport chain is when these electrons undergo a series of transfer between the chromosomes and carried of the electrons and is found in the rest of the mitochondria. One of the electrons carries in a coenzyme, in the other contain iron and called cytochromes. Each carried is at lower energy level than the previous one, and the energy that is released is used to form an ATP. This change produces 32 ATP molecules for each glucose molecule degraded, which plus 2 ATP molecules from the glycolysis and 2 more from the citric acid cycle. There are net gain to the 36 ATP molecules for the H-glucose is degraded into the dioxide of carbon and water.
Fermentation is another anaerobic, not oxygen-requiring pathway for breaking down glucose, one that's performed by many types of organisms and cells. In fermentation, the only energy extraction pathway is glycolysis, with one or two extra reactions taken on at the end. Fermentation is a chemical process that involves the anaerobic without oxygen transformation of an organic substrate into simpler organic compounds which cannot be metabolized downstream by enzymatic system without the intervention of oxygen. It is carried out by different enzymes and is normally observed in microorganisms such as molds, yeast, or bacteria, which produce a series of secondary products that man has used for commercial purposes for many centuries. In a chemical reaction that takes place during fermentation, enzymes, proteins capable of accelerating different chemical reactions, hydrolyze their substrates and break them down, or digest them yielding simpler molecules and more assimilate nutrients, metabolically speaking. When breaking cannot occur either because of the absence of an external electron accept or because of some defects in the cellular respiratory chain, fermentation is catabolic pathway used to produce energy from glucose or other carbon source. In the case of glucose, for example, partial oxidation is carried out through the glycolytic pathway through which pyruvate ATP and NADH are produced. Under aerobic condition, pyruvate is therefore then oxidized when entering the Krebs cycle and products from this cycle enter the electron transport chain. NAD is also regenerated during this process, allowing the continuity of the glycolytic pathway to be maintained. When there is no oxygen that is in anaerobic provided derived from oxidative reaction or the other resultant organic compounds, suffer a reduction. This reduction allows the regeneration of NAD, a fundamental event for the fermentation process. The reduction of pyruvate or other oxidative product marks the beginning of the synthesis of waste products, which can be alcohols, gases, or organic acids, which are excreted into the extracellular medium. And this image explains the glycolysis. The generation of pyruvate through the process of glycolysis is the first step in fermentation. Enzymes are proteins which act as catalysts. Enzymes lower the energy required for a reaction to occur without being used of the reaction. Many types of industries to aid the generation of their products utilize enzymes. Examples of these products are cheese, alcohol, and bread. Fermentation is a method of generating enzymes for industrial purposes. Fermentation involves the use of the microorganisms like bacteria and yeast to produce the enzymes. Enzymes have been used for thousands of years to produce food and beverage, such as cheese, yogurt, beer, and wine. Yeast is a fungus whose enzymes aid the breakdown of glucose into ethanol and carbon dioxide anaerobically. The enzymes in yeast break down sugar. glucose into alcohol, ethanol, and carbon dioxide gas. And this is the formulas that use. This reaction which takes place in the absence of oxygen is called fermentation. Fermentation works best when the gas and glucose solution is when warm. Enzymes will also become ineffective if the temperature complete oxidation of 1 mole of glucose to carbon dioxide, CO2, and water under aerobic conditions generates 38 moles of ATP. Fermentation produces between 1 and 3 moles of ATP per mole of glucose consumed.
Similarities between photosynthesis, fermentation, and cellular respiration are as follows. Cellular respiration and fermentation are processes that break down food and convert the chemical energy stored in the food to ATP molecules. Cellular respiration and fermentation begin with glycolysis and convert glucose to pyruvate. The energy molecules ATP are needed to the operation of these processes, and both of them use substrate level phosphorylated to generate ATP. Fermentation and cellular respiration start in the cytoplasm. Photosynthesis and cellular respiration involve in production of energy and the exchange of gases. Photosynthesis and cellular respiration take place in the cell organ, which was considered as endosymbiotic organism. They are chloroplast and mitochondria. Photosynthesis takes place in the chloroplast, whereas respiration takes place in the mitochondria.
All of these processes make energy or food to make more energy happen within the cell and neither create sugar or use sugar.
Difference between photosynthesis, fermentation, and cellular respiration. The fermentation process in cells releases about 2 energy units, whereas cellular respiration releases a total of about 38 energy units. Cellular respiration uses oxygen in the chemical reaction that releases energy from food, whereas fermentation occurs in an anaerobic or oxygen-depleted environment. Because fermentation doesn't use oxygen, the sugar molecule doesn't break down completely and so releases less energy. Photosynthesis has an anabolic metabolic process where carbon dioxide, ATP energy, and nicotinamide adenine dinucleotide phosphate are used to boil glucose molecules, while cellular respiration has a catabolic metabolic process where glucose is broken down to produce carbon dioxide and energy in the form of ATP, nicotinamide adenine dinucleotide phosphate, and flavin adenine.
Cellular respiration occurs in all living organisms, but photosynthesis only in plants, algae, and photosynthetic bacteria. Photosynthesis occurs in the chloroplast of the plant cell, and in cell respiration glycolysis occurs in the cytoplasm, while the mitochondria is the site of the Krebs cycle and electron transport chain.